So far, the adhesion molecules you have talked about, they require calcium for normal functioning. And now let's talk about certain molecules, adhesion molecules, that can function without the calcium ion requirement. Calcium independent cell-cell adhesion proteins basically belong to a large and ancient immunoglobulin superfamily of proteins. They all contain at least one Ig-like domain, which is characteristic of antibody molecules. One of the better studied examples of this category of molecules is NCAM, or neuronal cell adhesion molecule. It binds cells together by homophilic mechanism. We have discussed this previously also. Let me mention this again. Homophilic mechanism means two similar molecules are attached to each other. They're two similar molecules, same molecules are sticking with each other. Heterophilic, one molecule is different, the other one is also different, so that is heterophilic. So in this diagram, you can see these are the Ig domains. We have already seen Ig-like domains in cadherin molecules, but cadherin molecules require that a calcium ion binds the little groove which is formed between the two Ig domains. This molecule does not require calcium. Cadherin and Ig family members are frequently expressed on the same cells. Cadherin-mediated adhesions are much stronger and largely responsible for holding cells together. So these gain type of interactions, these NCAM interactions, they are more in the category of transient interactions, or the weak interactions, rather than strong interactions that we have seen are characteristics of cadherin molecules. So holding, this plays a important role. NCAM plays an important role in segregating cell collectives into discrete tissue and maintaining tissue integrity. NCAM and other members of Ig family seem to contribute more to the fine-tuning of adhesive interactions during development and regeneration. So again, these are more sort of transient interactions. And also, I would like to mention that not all NCAM-like molecules, they mediate interaction. They can also have repulsive effect also. For example, they can be heavily attached to sialic acid, which is a negatively charged molecule. Rather than attracting these two cells expressing this type of NCAM-like molecule, they will be repelling each other. Other example, formation of islands of Langerhans requires cell aggregation followed by cell sorting. Cadherin inhibition prevents cell aggregation and islet formation. Loss of NCAM basically just impairs the cell sorting process, so the islands that are formed, islands of Langerhans that are formed, are disorganized. The islands of Langerhans are basically regions of pancreas that perform an endocrine function, and they're about 45% of the total pancreatic volume. You may have heard of insulin. That is a hormone which is produced by these cells which are present in islands of Langerhans. NCAM-like protein called facilin 3 or FAS3 is expressed transiently on some motor neurons as well as on some muscle cells they normally innervate. If this FAS3 is genetically removed from these neurons, so everything else is constant, we just don't have these cells making FAS3 molecules. They fail to recognize their targets, which in this case are the muscle cells, and they do not synapse with them. Conversely, for example, if the motor neurons that do not express this type of adhesion molecule, if we make them to express or make FAS3 protein, these neurons which normally do not interact or make synapses with muscles expressing FAS3, now these neurons will start interacting and forming synapses with the muscles which are expressing FAS3. So again, this is a homophilic matchmaking sort of interaction which results in cell adhesion. So this is basically a special group of molecules that we just talked about, fine-tuning the cellular interactions rather than permanently sealing cells together.